Welcome to our explainer. It's wild to think about what actually happens when you just lift a glass of water or take a simple step forward. It feels totally effortless, right? But underneath the surface, it is literally an absolute marvel of biological engineering. Today, we are going to demystify the mechanics of movement. We are taking a powers of 10 journey together, starting way out at the macro, systemic level of your entire body, zooming all the way down to the microscopic behavior of a single protein, and then zooming right back out again. It's a beautifully structured system, and honestly, by the end of this, you are never going to look at your own movements the same way again.
Okay, let's dive right into this roadmap. First, we'll look at the body's systemic network. Then we're zooming in on muscular anatomy. After that, we hit the sarcomeres, the contraction headquarters. Next up is the sliding filament theory, followed by excitation, the nervous connection, and finally, movement and homeostasis. We are going to safely guide you through all this complex microscopic chemistry and back out to the big picture, I promise. Section 1. The body's systemic network.
To truly understand movement, we have to start at the absolute highest zoom level, the complete human organism. Your body is an incredibly complex network made up of 11 remarkable organ systems. You've got the integumentaries, skeletal, muscular, nervous, cardiovascular, lymphatic, respiratory, digestive, urinary, endocrine, and reproductive systems. All 11 of these are constantly collaborating just to maintain homeostasis, that vital internal balance required to keep you alive. But when it comes to generating force and actual locomotion, the muscular system is our absolute star player today.
Now, anatomists use a few different approaches to study the body, and one of the most effective is systemic anatomy. Basically, instead of just looking at the arm or the leg in isolation, we look at a specific body system as one whole functional network. So when we study the muscular system this way, we're actually looking at over 700 skeletal muscles in your body as one collective, unified engine, completely regardless of where they are physically located. Section 2. Zooming in. Muscular anatomy. Okay, so we've got our systemic context. Now we're zooming right past the organ level and going straight into the tissue level. And this brilliantly illustrates how the macroanatomy of a skeletal muscle is essentially a layered Russian nesting doll. On the outside, the whole muscle is wrapped in a connective tissue called the epimysium. Open that up, and you find bundles called fascicles, which are surrounded by the perimysium. Crack open a fascicle, and you finally reach the individual muscle fibers, or muscle cells, and each one of those is encased in its own endomysium. This continuous network of connective tissue is crucial because it ultimately links right to your tendons, transferring the tension generated deep inside the cell straight to your bones.
Section 3. Sarcomeres, the contraction headquarters.
Plunging deep inside that single muscle fiber, way past the normal cellular organelles, we find hundreds of myofibrils. And these are totally packed with two primary protein filaments. On one side, we have the thick filaments made of a protein called myosin. It looks a bit like a golf club with two globular heads and a long tail. Then, on the other side, we have the thin filaments, primarily made of actin. But here's the catch. Actin isn't acting alone. It's surrounded by two regulatory proteins called tropomyosin and troponin. Tropomyosin basically acts like a physical guard, covering up specific binding sites on the actin strand. And troponin? That is the lock attached to the guard, just waiting for a very specific chemical signal to pull that guard away. Now, what's really interesting about this is how these filaments are physically organized. They're not just floating around at random. They're arranged in this highly overlapping scaffolding. This repeating functional unit is called a sarcomere. By definition, a sarcomere is the highly organized space between two Z-discs. You can see those zigzag vertical lines right at the edges. This arrangement is exactly what creates that distinctive striated or banded appearance of skeletal muscle. And this structure is the absolute foundation for how a muscle physically shortens.
Section 4. The sliding filament theory.
So all of this crazy structure exists for one main reason: the rapid-fire sequence of how contraction actually happens. Let's break it down. First, depolarization and calcium release. An electrical impulse causes the cell's internal storage vault to flood the area with calcium ions. Second, cross-bridge formation. That calcium binds to troponin, which shifts the tropomyosin guard out of the way, allowing myosin to finally grab on to the actin. Third, the sliding mechanism. The myosin heads forcefully swivel, sliding the actin filament right along. And fourth, sarcomere shortening. Because the actin is anchored to the ends of the sarcomere, the entire unit physically shortens. Now, to fuel this movement, the myosin head requires a molecule of cellular energy called ATP. When ATP breaks down, that energy causes the myosin head to physically swivel. I love this visual metaphor. Think of it exactly like an oar propelling a rowboat through water.
Grab, pull, release, reset. Grab, pull, release, reset. As long as there is calcium and ATP available, millions of these tiny oars are furiously rowing, dragging the actin filaments toward the center. So the absolutely crucial point here is this. The protein filaments themselves, the actin and myosin, do not shrink. They don't change their own length at all. They merely slide past one another. The Z discs get pulled closer together, the I bands get shorter, and that central H zone practically disappears. This sliding action shortens millions of individual sarcomeres simultaneously, which in turn shortens the entire whole muscle.
Section 5. Excitation, the nervous connection.
Let's move on and see how this builds, because we really have to ask, where does the initial spark for all this intense molecular rowing actually come from? Muscle fibers don't just decide to contract on their own. The nervous system has to tag in. A motor neuron sends an electrical action potential down to the muscle. Right at the neuromuscular junction, it releases a neurotransmitter called acetylcholine. That chemical signal triggers a wave of depolarization across the muscle cell membrane, diving deep into the cell through these specialized tunnels called T-tubules. This is the exact signal that commands the cell to release the stored calcium and begin the whole rowing process.
Biologists actually call this incredible partnership excitation-contraction coupling. As this quote highlights, it links the depolarization of the muscle cell membrane to the initiation of myocyte contraction. It's a great reminder that without the electrical command from the nervous system, the muscular system is, quite literally, completely powerless.
Section 6. Movement and homeostasis.
Zooming all the way back out to the macroscopic level of the whole human organism, all that rapid molecular rowing requires massive amounts of ATP. And get this, here is a truly surprising fact. Only about 25% of the energy released from breaking down that ATP is actually used for the mechanical work of pulling the muscle. The other 75% is released purely as thermal energy. No way, right? But this immense heat generation is actually a crucial function of the muscular system. It is the primary way your body maintains its core internal temperature of 98.6 degrees Fahrenheit.
And how do the muscles even get all that ATP and calcium in the first place? Well, this is where looking at the larger context of organ systems, our systemic anatomy, really shines. The respiratory system pulls in oxygen, and the digestive system extracts nutrients from your food. The cardiovascular system acts as this high-speed transit network, pumping that oxygen and fuel directly to the muscle cells while carrying away carbon dioxide. Simultaneously, the urinary system is filtering the blood, meticulously maintaining the precise balance of water and electrolytes, like calcium, that your muscles desperately need to function.
It is just an awe-inspiring loop of biological dependency. From the whole 11 organ systems working together, down to the tiny myosin oars, and back up to the maintenance of your body temperature, it leaves us with a pretty provocative thought. What happens to the internal balance of our interconnected systems when even a single microscopic misfire disrupts this intricate chain of events? The delicacy of homeostasis is truly profound. Thank you so much for joining me on this deep dive into the mechanics of movement. Stay curious, keep exploring, and I'll catch you in the next explainer.